Hello, everybody. Just before we start our webinar, I would like to remind you that we'll record it and we'll share it after our meeting. So it's great that you are here. I can see that there's quite a lot of persons already here, but people are still joining. So we'll try to start slowly to give them chance to join us. I'm Wojtek Mróz, and in behalf of Eurosite Secretariat, I will host this meeting. And I hope that you all can sit comfortably and there's no any technical troubles. And before we go to the content and we introduce our today's speakers, I would like to tell you about rules of our webinar. So as I said, this webinar will be recorded and shared, and all of your questions you can send through the Q&A button, which is on your right hand down there, and you can write there any questions that are connected with our meeting. And also remember that your microphone and cameras are turned off. So this time, I see that we can have some technical troubles. Sorry. I would like ask our first speaker, Nigel Maxted from Birmingham University, to start with his presentation and to tell us more why we are here and what it's all about with these crop wild relatives. Hello, Nigel. Are you there?
Yes, I'm here.
Great.
So shall I start?
Yeah, please go on.
Okay. Right, my name is Nigel Maxted. I'm from the University of Birmingham in the UK, and I'm supposedly an expert on crop wild relative and landrace conservation. And you can see from the number of people included in the authorship of this presentation, I'm taking pieces of information from previous presentations I've had given with colleagues in the past. So the first point I wanted to make is that human population and climate change are changing how we think about agrobiodiversity conservation. Perhaps human population growth is not a significant issue in Europe, but it's a significant issue globally. And 70% is estimated by 2050, we need to increase food production by 70% to be able to feed the population in 2050. And that's 100% increase in developing countries. While at the same time, climate change is going to impact agricultural production and decrease production by 2% per decade. It's all very well giving figures, but I like to give this nice, what I think is a nice illustration of the climate change's impact. This is a study that was done in Oman in 2015, and we were looking at one species, alfalfa, Medicargo sativa. And you can see in 2015, 12%, the red areas, 12% of the country could actually grow alfalfa. And in the short term, now, 2020, it's actually increased to 17.4%. But if we look ahead to 2050, you can see by 2050, it's decreased dramatically to 2.3%. So if the predictions of climate change are correct, then we must have active conservation of genetic resources now before they disappear and they're unable to grow in their home environments. In terms of meeting the breeder's requirement to be able to sustain crop production in the future in light of climate change, we are going to require greater breadth of trait diversity. And that breadth of trait diversity comes from crop wild relatives. There are, because these are wild species, that haven't gone through the bottleneck of domestication, they still have a very wide range of adaptive traits within them. They're tried and proven sources of traits that can be transferred into the crop, and very large numbers of traits have been transferred for about the top 15 global crops, though obviously the potential there is for transferring into all crops. And with technology advances in recent years, trait transfer is becoming easier and easier. So what are crop wild relatives? So they're the wild species, like any other wild species. They're the ancestors of crops, and so potentially they have value now as gene donors. And as I just said, because they haven't gone through the bottleneck of domestication, they have a lot of novel genetic diversity that can be used. So broadly defined, a crop wild relative is any species or taxon in the same genus as a crop. That's the broad definition. And we can become much more precise if we look at gene pool concepts and how easy it is to cross the wild species, the wild crop wild relative with the crop itself. But generally, we think of crop wild relatives as any wild species in the same genus as a crop species. And just to show how many there are, this is the sum of the wild relatives of wheat, the crop wild relatives on the left-hand column, and on the right-hand column are the different traits that have been transferred into wheat from that crop wild relative. So the point I'm making here is that there are a large number of crop wild relatives and a very large number of traits that have been transferred historically from the crop wild relative to the crop. And the value of these, astoundingly, the value is given in a paper first published in 1997. The annual value of using crop wild relatives in breeding was $115 billion per year. And that figure was also coincidentally about the same figure that was given for 29 crops in 2013. So 115 billion, a very large amount of annual value generated from crop wild relative species. Now you think species that are that valuable would be very well conserved and not threatened. But if we look systematically at this, there was a review done in 2011 of 572 European crop wild relatives, and it was found among those 16% of them were threatened with extinction and 4% were critically endangered, meaning they were likely to go extinct in the very short time frame. So crop wild relatives are threatened. We must act now to conserve them, otherwise they'll be lost in the future. In terms of ex situ, in situ conservation, if we look at the number of wild taxa conserved in European gene banks, it's about 10.5%. And Castaneda, not looking at Europe, but looking at global conservation of crop wild relatives, found about a third of all crop wild relatives have no accessions at all in the gene banks. About a third have poorly conserved, so that's less than 10 accessions per species or taxon, and 72%, it was a high priority to collect further material. So they're not well conserved in terms of ex situ conservation. But the situation for in situ conservation is even worse. So there are very few, although they found in a lot of protected areas around the globe, they're passively conserved. In other words, they're present within the protected area, but they're not being actively managed to sustain that particular population. So there are only a handful of what we call genetic reserves within existing protected areas globally, where they're being actively monitored and managed to maintain the crop wild relative population. And I've given some examples of those here. But I would guess that none of these ones that do exist meet the standard suggested by Jose Uriondo, who's going to be speaking later. And the standard is they must be, there's a whole set of criteria, and not all of these in situ populations that exist today meet those existing standard criteria. There's a policy imperative to conserve crop wild relatives, and they're mentioned both in the CBD strategic plan, talks about wild relatives, their status in terms of conservation has been improved. And in the global strategy for plant conservation, it also talks about conservation of wild relatives. Both of those two priority actions are running out of time. They're both due in 2020, and we're reformulating those at the moment. So we're pretty certain that crop wild relatives will come out as an ongoing priority for conservation action. And they also obviously have impinge on the UN Sustainability Development Goals, particularly goals 1, 2, and 3, and most of all 2.5, which specifically talks about the conservation of agrobiodiversity, including crop wild relatives. So the breeders require greater diversity, greater trait diversity to be able to tackle issues like climate change and sustaining food production or increasing food production in the face of climate change. Ex situ, I would say, has done a very good job historically, but with the increased demand for diversity, will they be able to maintain this in the future? So now increasingly people are turning to in situ to provide some kind of complementary, additive conservation to build on top of what is already being done ex situ. And the idea of establishing a completely new set of protected areas globally or in Europe for conservation of crop wild relatives is unrealistic. It's not going to happen. We don't have the funding. So we want to establish crop wild relative genetic reserves within existing protected areas. Why? Because those existing protected areas have a long-term ethos for conservation. They're unlikely to suffer any hasty management changes. So there's long-term conservative values to the maintenance and management of the sites. It's relatively easy to make minor adjustments to the site management plan to cope with the monitoring of crop wild relatives. And as I said, to actually have new sites would be prohibitively costly. The other point I'd make in the European context is crop wild relatives, as you'll hear from Jose's talk later, appear in significant number in the Natura 2000 network sites that already are set up across Europe. So historically, there has been seen to be what might be considered two groups of people working on conservation. Those who work on ecological conservation, focusing primarily on habitats and ecosystems, and those working on genetic conservation and making the link between conservation and use. So the methodology we use for actually undertaking crop wild relative conservation is, first of all, we have the basic diversity of crop wild relatives or land races or other forms of genetic diversity. We actually plan the conservation, which is a process of comparing the diversity that exists in nature or on farm with the diversity that is actually conserved. And that gives us our gaps. The gaps become our priorities for conservation. And in terms of traditional ecological conservation, that's where it would finish. So the added value of genetic conservation is there is this link through to utilization of the conserved resource. So we have characterizing in evaluation of the resource and then utilization that adds to that final value. So that kind of analysis that I've just described there has been done at country level. And so this is an example of that kind of gap analysis for identifying sites across the UK for setting up, I think, the top 25 protected areas in the UK for crop wild relative conservation. And the one that is actively being set up at the moment is Site 6. And it was set up at the basis of Hannah Fielder's work here, one of my former PhD students. And that site is on the Lizard in Cornwall. And you can see there's a list there of the major crop wild relatives found in that location. So wild alliums, wild asparagus, wild beeters, carrots, radish, and various trifolium species. And you can see the populations found round the coastal border of the Lizard Peninsula. And then the same approach has been taken at a national level, various countries across Europe, and then globally outside of Europe. So basically the methodology is sound. And I want to show it can work not only at national level. Oh, so just before I go on to talking about global level. So I want to make another point before I went on to that, and that's management. So once you've identified your sites as being rich in crop wild relatives, you might imagine you have to radically change the management of the site. That is not the case. So this is looking at wild chives on the Lizard Peninsula in Cornwall. The management required is the management they actually have at the site. You have a healthy population of your target crop wild relative, and it's being managed to actually make that healthy population. So there is no change to the management required. So all we're doing is making sure we do not change the management into the future. It's sustaining the current management is the way of maintaining it. So the only change to the management of the site is to actively monitor those target populations to make sure they're still there into the future. So as I said, I wanted to show that we can use the same analysis for looking at other scales as well. So this is a global analysis undertaken by the Global Crop Diversity Trust with funding from the Norwegian government and various partners. And they identified a species richness map for 1,394. These are the global priority crop wild relatives, which are related to 194 crops. And you can see here, you've got a very red area or high concentration area around the Mediterranean. This is looking at the ex situ conservation priorities. So this is looking at that richness map and then combining it with a map of concentrations that have already been sampled and put in gene banks, and the gaps, identifying the gaps. So these are the gaps shown here. So you can see, for instance, Portugal, Turkey, Indonesia, parts of Brazil are countries where we need to have high levels of future conservation. And this is looking at in situ conservation. So this is the top 150 sites around the world, looking at the same 1,394 species. And so each of the 150 sites, we have 5 sites per species. They're selected to maximize genetic diversity, and each site is been climate change proof. So in other words, the site will have a long-term future. It won't disappear because of climate change in the next 10 years. So these are the top 150 sites around the world. And this is a close-up of the ones in Europe to show you that there is a very high concentration, a global concentration of crop wild relative diversity in Europe. And you can see the sites we've already identified here. So what we're talking about, what our proposal is to build a mutually beneficial collaboration between us working on crop wild relative or land-raised conservation and protected area managers. This involves complementary conservation as defined by Ford, Lloyd and Maxted, as including both in situ and ex situ conservation. So some protected area based work, but also some sampling and putting material in the gene bank. Currently, the amount of funding that goes into crop wild relative and land-raised conservation is primarily for ex situ. So what we want to do is complement that by adding additional funding to do in situ conservation and then linking that in situ conservation to use. Because we know that ex situ alone is unlikely to be able to meet the future needs of breeders. And in Europe, the ideal protected area network we would work with is the Natura 2000 network, which, as I said, Jose will talk about later on. So how is it mutually beneficial for us in the agrobiodiversity community? There is a lack of systematic crop wild relative conservation currently. We don't have much or any really in terms of in situ action, but we can't set up in situ conservation on our own. We have to work with collaborators, and the collaborators are the people who are already managing the in situ sites. So the benefit to us is systematic crop wild relative conservation and greater diversity available for use. In terms of benefit to the protected areas, the problem is it's quite difficult to show a link between protected area conservation and the UN Sustainable Development Goals. And there is this insecurity of funding potentially associated with existing protected areas. So if we can add the additional dimension that you're conserving resources to feed humankind in the future, then that is an additional value, an additional ecosystem service. And it comes at a minimal cost. As I explained, we're not talking about changing the overall management sites, because the sites we're identifying are ones that already have healthy populations of crop wild relatives within them. So Farmers Pride, I wanted to talk a bit about Farmers Pride. So we're doing this in the context of Farmers Pride, a Horizon 2020 project, which has 44 partners, including Eurosite, And one of our deliverables in that project is, by the end of the project, to set up a European in situ conservation network of stakes and stakeholders to actually manage the diversity of crop wild relatives and land races. And that would be, we hope, be within the context of the Natura 2000 network. I also wanted to mention the second Horizon 2020 project, which is GenRes Bridge, which is working on the more of an integration of plant genetic resources, forestry genetic resources, and domesticated animal genetic resources conservation, and say there is a future benefit for doing forestry or animal genetic resources conservation also within protected area networks. So there may be an additional link in the future for Natura 2000 there as well. So I think this has a lot of potential for the future. In terms of nominating of sites, we're at the stage in our project now where we're asking people who are managing sites that have, we know, crop wild relative or landrace populations within them of significance, to take our survey and help us nominate the best sites in Europe to become part of the network we're setting up. And you can see the website is there, you've got the address, and you'll receive a copy of the presentation afterwards, so you don't have to copy it down, or you can get it via our website anyway. So final slide. So I think we have a unique opportunity at this time. The unique opportunity is to double the current diversity available to farmers and breeders. When I say double, I mean double in the sense that we're complementing what is already being done in terms of ex situ by building this in situ network, and the material contained within the in situ network, if it's working effectively, can actually double the amount of diversity that's available for breeders to use in their climate change mitigation or to help with the generation of bigger, better crops. We're starting in Europe, but we hope eventually to build a global network of sites, which will be called the Vavilov Network, named after the scientist, the Russian scientist, who first started to talk systematically about conservation of crop wild relative and landrace diversity. It's important for us, we're talking about complementary conservation, so making sure that we have this link between ex situ and in situ conservation, and then the link through to use. So just as historically breeders have always accessed the material via a gene bank, so they must be able to get material out of in situ reserves into their breeding programs. And the fact is, we need to take action now. It's time for no regrets action. We need to be active now, but we're lucky in that we have the public on our side. And as a final point, I found this last year talking about the number of EU citizens that have that believe we have responsibility as the population of Europe for conservation of nature. I'm sure if you added to that by conserving nature, you're also securing the food that's available for your great-grandchildren to eat, then the figure would have been even higher. Thank you very much. That's my introductory talk, and I'm passing back now to the chair.
Thank you, Nigel. It was really great and impressive presentation. Thanks a lot. And now we still have something like 5 minutes for questions directly to Nigel. And I would like to pass the floor to Joana Brem from Portuguese National Gene Bank, and she will help us with Q&A box, because I've seen that she found quite a lot of interesting questions. Joana, are you there?
Yes, I am. Thank you. Thank you, Nigel. We have a few questions here. And one from Luigi Guarino is, have there been any examples of reintroduction of crop wild relatives?
Reintroduction from extinct populations, does he mean?
I think in general, if
so,
perhaps it threatens plant species, threatens crop wild relatives that need to be reintroduced, I presume.
Yes. So there is an example of a species which I found myself called Vicia calicensis, which was found near Tel Kelek in Syria. And because of a mistake, the local population was virtually wiped out by the local community by accident. And so we actually took material from the Akada gene bank, we took seed back to that location and replanted it. That was probably about 15 years ago. Obviously, I haven't been there recently to check how the populations are doing. One hopes they're doing well.
Yes. Okay. I hope Luigi is happy with that answer. At least we have that example. Second question by Juozas Labukas from Lithuania. He says, Dear Nigel, are there any monitoring and or management plans prepared or implemented for the genetic reserves in the UK, although they are not officially recognized so far?
Yes, so we have only one site which has crop wild relatives at this stage included within its remit, and that's the site I was talking about, the lizard in Cornwall. But actually, I'm very excited to tell you that we're doing a review of all national nature reserves within the UK and looking at all of them for potential, whether or not they can become genetic reserves for crop wild relative conservation. So in which case we will have all of those management plans amended to make sure that at least monitoring is included within the management plan. So I'm very excited. It's a new initiative which is just starting now.
And for how long will that be funded, I presume?
Because of the lockdown, unfortunately, it's somewhat up in the air. So that's an honest answer. Before the lockdown, we had it all planned and it should have been at least well underway by now. But my guess is it'll start after lockdown finishes.
Okay. We have another question by Christopher Cockle. He says, please, can you explain the process of prioritization in terms of what we concentrate our efforts on?
So if you ask me if we're dealing with all crop wild relatives, then obviously the ones we should be focusing on, because we have this difference between, as I said, genetic conservation and ecological conservation. It's not just about conserving populations, it's making sure the populations are available for use. And so the material that is most widely available for use will be the ones that are most closely related. So if you think about it in terms of gene pool concepts, it's the gene pool 1B. So it's the species which they're either subspecies or very, very closely related to the crop. And there is no breeding barrier between the crop and the wild relative. So that would be our first priority. And then the second priority would be gene pool 2 species, where there is some slight breeding barrier and you may have to overcome that using breeding techniques. So you're basically focusing on the ones that are most useful to the breeder or the farmer.
Thank you. Another question by Julian Hosking. Whether you agree with the following idea that crop wild relative conservation needs to be integrated into all Natura 2000 sites and their management plans?
I'd like to agree with Julian, but the only thing I would slightly say is that maybe it's a big ask to ask that to happen within all Natura 2000 sites. So I would like to start off to have a kind of proof of concept. If we started off at saying we were aiming for 100, 150 sites across Europe, and then if that proves successful, extend that out to the other sites, that would be a really good start, I think, and a sensible way to approach it. But in principle, yes, I don't see any reason why all of the Natura 2000 sites shouldn't be able to do active conservation of crop wild relatives within their boundaries.
He also adds 2 other sentences to see whether you agree with this, that all Natura 2000 sites need to provide seed and other samples of their crop wild relatives at regular intervals to national seed banks with open ABS access. And the crop wild relatives are also useful for many other uses beyond food for people.
Right, so answer the first one first. And I think this is the one that's really challenging for us in terms of in situ conservation. Because historically, as I said, breeders have got all their material from gene banks, and gene banks are set up to supply this material. So my initial thought was that in situ sites would provide the material direct to gene banks. And I think, frankly, that's not going to work, because it is onerous and it's time consuming to do that. So it would be much better if, as part of conserving crop wild relatives, you make samples available to an associated, a linked gene bank, and you get the material from the gene bank, and then it would be a normal supply of the gene bank. But then I'm sure people listening to this who work in gene banks might be saying, well, look, that requires a lot of additional work, and we don't have the funding to do that. And so I think you would have to have the crop wild relatives under a slightly different regime within the gene bank. And so there are various things within the gene bank that cost a lot of money, and one is regeneration. So I think you would not regenerate material from in situ within the gene bank. You would just go back to the in situ site and take fresh samples. So that way you're getting over the main cost of in situ conservation for the gene bank. And they would be happy, I hope, to then supply the material to the users. So it's building up a relationship. It's an active relationship between the ex situ, in situ, and the user community, all working together to conserve and meet the needs that we basically need for the future.
Wojtek, do we have more time for answers and questions? I think we
can ask the last question to Nigel now.
Okay, so there's someone who says, thanks, Nigel. The biggest problem is having many crop wild relatives occur naturally in disturbed habitats like roadsides. So it's difficult to apply management approach like Lactuca and Daucus.
Right, okay, so that is true. Many crop wild relatives exist in ruderal reedy species, and so do exist in roadsides, edges of cultivation, etc. But a lot of them are also found in the Natura 2000 network. And Natura 2000 network may itself include roadsides. So we can have conservation within those locations within the Natura 2000 sites. But I also, in parallel, would like to start looking more actively at conservation outside of protected area networks. Though there is no precedent for very few precedents for doing this approach. So I think this requires much more research and thinking through. And so in the short term, working with an existing protected areas has to be our priority. It's the logical way of doing it in the short term.
And in fact, just to add to that, there's someone who says that, who asks, because crop wild relative species are also found in these marginal zones, particularly near agricultural sites, whether you will be supporting the idea of engaging with farmers close to these existing sites as a priority.
Yes, but you have to realize that the Natura 2000 network is not, they're not all pristine climax communities. A lot of them are sites that do include farmed land. And so you'll be working with the farmers within those sites to actually conserve material in those locations. So I don't see there being a contradiction there.
Time for one more question, Wojtek?
Yes, I think the very last one.
Okay. So Alvina Vagian asks whether there are any special indicators for monitoring crop wild relative biodiversity in protected areas.
Yes, I think the indicators that are usually used are demographic. So it's counting the number of individuals for a particular crop wild relative species. I mean, I would like to say that we're able to do genetic diversity monitoring, but I still think in terms of regular in situ site expenditure, it's beyond us to have routine, regular genetic diversity management. So in the absence of being able to do that, and that's got nothing to do with the Natura 2000 network, that's true of all protected areas. In the absence of that, then it's just simple, straightforward demographic monitoring, which is exactly what protected area managers have been doing for as long as there have been protected areas, the last 100 years.
Okay, thank you very much, Nigel.
Thank you. Good questions. Thank you.
Thanks a lot, Nigel, for your presentation and explanation and your help, Joanna. We'll ask for your help later on after the next presentation. Thanks a lot. And now I would like to introduce our next speaker. As you can see on our program, it will be Sheila Kell from Birmingham University. And she has been working international in the field of plant genetic resources for many, many years, and he has particular expertise in crop wild relatives. In addition to her roles, she's also a chair of the genetic resources section of the European Association for Research and Plant Breeding, EUCARPIA, and editor-in-chief in the journal Plant Genetic Resources, and many, many others interesting roles. But I will just pass her my virtual mic. Sheila, are you there?
I'm here, but I cannot show my screen at the moment, Wojciech.
We can see your screen with, yes?
You can see me?
Yeah, we can see your presentation.
No, but can you see me? I can't share my camera.
Just a moment, we'll check it.
Okay, great. Thank you.
Thanks a lot.
Okay, thank you very much, Wojciech, for the nice introduction. Nigel has given an overview. He's explained what a crop wild relative is, why these species are important, and given the global context, and also emphasized the importance of bringing the agrobiodiversity and protected area communities together. So now I'm going to focus in on Europe and look at what crop wild relative, important crop wild relative diversity exists in the region, and explain the process of selecting priority species for conservation action. So I hope this should answer the question of Chris Cockle, which Nigel has already partially answered. So Europe is an important centre of diversity of many crops and their wild relatives, and these species are potential genetic resources for crop improvement, and thus important for food, nutrition, and economic security. There are many crops in the region which have significant crop wild relative diversity in the region. These include major crops such as wheat, barley, oat, sugar beet, cabbage, and several other brassica crops, onion and other alliums, asparagus, lettuce, and apple, just to mention a few. And then also a lot of diversity of wild diversity of forage and fodder crops, including annual meadowgrass, white clover, alfalfa, and common vetch. These are just a few examples. So we have a lot of important diversity of crop wild relatives in Europe. So first of all, I wanted to, before talking about the selection of priority species, I wanted to mention that this work that we're doing is ongoing work on the development of a crop wild relative conservation strategy for Europe. And at the moment, we're focusing on this in the Farmers' Pride project, but it relates to a concept for in situ conservation of crop wild relatives in Europe that was adopted by the European Cooperative Programme for Plant Genetic Resources in 2015. And central to that concept is this two-pronged approach to crop wild relative conservation planning, which you can see in the figure on the right. Now, I'm not going to go into detail on this, but it's just to say, to emphasize that Nigel mentioned national crop wild relative conservation planning. Of course, we have to undertake a process at national level because countries have sovereignty over their genetic resources, but it's also important to take a regional approach to conservation planning to ensure that we are conserving the maximum crop wild relative diversity and that nothing is being missed by taking a national approach. And if you want to read about this concept, you can see the link there to this document, which is available online. So what I'm going to be talking about now is, as I say, this regional approach to crop wild relative conservation planning. And so the selection of target species for conservation action of species which are important for the region as a whole. So the process is based on 3 main criteria, and this is a model that was published in 2017 for a pragmatic and systematic approach to crop wild relative prioritization. So the first criterion is the socio-economic value of crops, which may seem to be fairly obvious, but it's important to emphasize that these are wild plant species like any other plant species, but their value lies in their relationship to crops. So they're distinguished from other wild plant species by this relationship and potential use for crop improvement. The second criterion, which Nigel has already mentioned, is the utilization potential of those species related to those crops. And then the third criterion is the relative threat status of those wild relatives related to those highly important crops. And I'm going to talk through the application of these 3 criteria for the prioritization of Europe's crop wild relatives. So again, it may seem obvious that we have to take into account human food crops when we're prioritizing crop wild relative diversity, obviously important for food security and nutrition security. And one indicator of important crops is to look at FAO stat data on the production value. And this shows 46 crops or crop groups that are cultivated in Europe, which have crop wild relative diversity in the region. And the value here is in billions of US dollars. So even sugarcane, which is not extensively cultivated in the region, it has a very high value. So on this basis, these are the human food crop groups that are prioritized, and plus human food crops not cultivated in Europe, but have crop wild relatives in the region. So an example is quinoa, which I know actually we are starting to cultivate it, and there seems to be some initiative to find breed varieties suitable for cultivation in Europe, but it's not extensively cultivated and there are no production value data. But quinoa does have wild relative diversity in the region. And it's important that we conserve that diversity because of the interdependence of countries and regions on genetic resources. So we have a responsibility to conserve the crop wild relative diversity in Europe, which may not be of immediate value to us as a region, but may be of value to other parts of the world. So the second group are animal food crops, again, because they're important for food and nutrition security. And to select the crop species in this group, I looked at species listed in Council Directive on the marketing of fodder plant species, species listed in the EU Common Catalogue of Varieties of Agricultural Plant Species, and species listed in Annex 1 of the International Treaty on Plant Genetic Resources for Food and Agriculture, which is a list of crops and crop gene pools which are of global importance for food security and interdependence. And any taxa that were not already listed from those 3 sources, for which seed production data are provided by the European Seed Certification Agencies Association. And this is just to show, it's not important that you can see the details of these charts, but just to show some of the indicators of the value of these animal food crops. So we can look, for example, at the numbers of registered varieties in the Common Catalogue. We can look at fodder seed production acreage and fodder certified seed quantities. And although some of these are obviously more important in terms of their economic value than others, it's important to conserve as many of these species as we can. So we prioritize all these crops to ensure that we conserve the maximum diversity of all of these important crops. So going back to the model, we know that in Europe and the Mediterranean region, we have a huge number of species which we can consider to be crop wild relatives, broadly speaking. So this is based on a broad definition of a crop wild relative, which, as Nigel said, can be considered as any taxon within the same genus as a crop species, but also on a broad definition of a crop. So the crop wild relative catalogue for Europe and the Mediterranean includes not only human and animal food crops, but also crops with other uses such as industrial, medicinal, ornamental and forestry. So after applying criterion 1, the socio-economic value of crops, we have 210 human and animal food crops which have crop wild relative diversity in Europe. And Turkey is written in brackets because in the Farmers' Pride project, Turkey is a partner and also is an important country for crop wild relative diversity. And these 210 crops occur in 102 genera. Now, if we look at the taxa within those genera, taking a broad definition of a crop wild relative, we'd be looking at trying to conserve more than 4,000 species. So clearly it's not possible to prioritize to conserve that many species. So we have to select which ones are most important for conservation action. So we look at criterion 2, which is utilization potential. And as Nigel already noted, we want to prioritize the closest wild relatives initially. These are the taxa which are most usable in conventional plant breeding and generally have been used more widely for that reason. But we don't want to ignore the tertiary wild relatives. Although tertiary or more distant wild relatives are more difficult to use in conventional plant breeding, they are important and they have been used, and they are still extensively used and increasingly used, in fact, using sophisticated techniques. And so we want to make sure that we prioritize those species that have known use or potential use for crop improvement, not just the closest wild relatives. And then Nigel also, I think, mentioned the taxon group concept. This is just a proxy in cases where we don't have the information required to apply the gene pool concept. The taxon group concept is based on the taxonomic hierarchy as an alternative. And this is just to emphasize the importance of these more distant tertiary wild relatives. These are examples of traits in tertiary European priority crop wild relatives that have already been used in crop improvement. And you can see there that most of the traits are for pest and disease resistance, but there are also traits there for abiotic stress tolerance. So for example, soil salinity, cold and frost, and also other traits such as improved aroma and day length in strawberry. So these, again, as I say, are tertiary wild relatives. It's just to emphasize that these species, these more distantly related species, are extensively used, and so it's important that we also conserve them. And as Nigel mentioned earlier on, breeders are looking to more exotic sources of diversity in order to breed crops resistant or adapted to climate change and the changing environment and uncertain climatic conditions that we're facing. And it's important to say that the reason that we are able to apply this criterion utilization potential relatively easily now is a huge amount of work that's been undertaken. These are two main sources of information. One is the work undertaken by the USDA Agricultural Research Service, and there's a searchable database online via the Genetic Resources Information Network. where there's a huge number of crops there, and you can look up the gene pool concepts of those crops. And then the work that was undertaken by Nigel's PhD student Holly Vincent and published in 2013. She created the Harlan and DeVette Crop Wild Relative Inventory, and that is available via the Crop Trust website and is constantly updated. So going back to the model, we've applied Criterion 2, and this reduces our number of taxa to 793. So we have, based on their utilization potential, we have 418 species related to those important 210 human and animal food crops. Now, as I've just stressed, it's important that we don't ignore the tertiary wild relatives, but of course we can't hope to plan conservation of all of them because there's a huge number. So a pragmatic approach is just to select tertiary wild relatives that are under threat or known to be threatened, in addition to those that are of utilization potential. So in the case of the European species, as Nigel mentioned, there was a big project, the first major initiative to undertake RED list assessments of crop wild relatives. And this was published, the assessments were published in 2010. And so this is the best information we currently have. And so based on this work, we add another 67 species which are either threatened or near threatened, and 3 subspecies. So we have a total final list of priority taxa comprising 863 taxa in 485 species. So these are species which are related to important, socio-economically important crops. They have enormous utilization potential, and some of them are also threatened or near threatened, so need particular attention. So just a final few slides. I want to just emphasize the importance and the value of these species in Europe. And this chart shows the related crop use categories of these taxa. And you can see that the highest number of taxa are related to fodder and forage species, crop species, and then we have cereals, vegetables, fruits, and so on. The total number is 1,061 because many species are related to more than one crop. So the same species may be related to a fodder crop, but also related to a cereal, for example. So it has multiple potential use. Then we also know that out of these priority 485 species, 169 contain known confirmed or traits with confirmed or potential use. So confirmed use means that they've been used already for crop improvement. Potential use means that they've been characterized and we know that the traits exist, but they haven't actually yet been used in crop improvement. So 225 traits at least that we know that are useful for crop improvement in 35% of these priority species, which is very reassuring. We know that at least for 35% of these species, we already know they have this value. And it also shows that there's enormous potential in the remaining 65% of species for which we don't yet know whether they have any useful traits, but once characterized in the future, there may be of enormous potential. So what do we know about the red list status of these 485 species? So again, this is based on the best information we have at regional level. So this is based on European regional assessments of crop wild relatives, the ones published in 2010. And at that time, we assessed a larger number of species related to a smaller number of crops. The reason for that was because at that time, 10 years ago, we didn't have the information available on the utilization potential, the application of the gene pool and taxon group concepts, only for a few of the crops. And so we assessed species related to 25 human food crops and a number of forage species as well. So based on this, what do we know about our current list of 485 species? We know that at least 17% are threatened or near threatened. We know that 6% are data deficient. That means there wasn't sufficient information at population level to actually even assess them. A dearth of information. We know that 33% are assessed as least concern, but it's really important to say that even though they're perhaps a bit more widespread, according to the IUCN criteria, they're least concern. It doesn't mean that we don't need to conserve them. We do need to conserve these species. The point of conserving crop wild relatives is to conserve diversity within and between populations. And so it's important that we do focus even on widespread species to make sure we're conserving representative samples. The other reason is because some of these species may be threatened locally or nationally. These assessments are at regional level. I'm just coming up to my last slide in a moment, Wojtek, just to see you there putting some pressure on. And then 43% at the moment are not evaluated. And as I say, that's because we assessed a smaller number of gene pools in that project. Because these assessments as well are now 10 years out of date, of course it would be extremely pertinent, timely now to undertake another red listing project for these important species. So just one final slide. This is just to emphasize that there are 86 globally threatened or near threatened priority species related to 38 crops. So these are species which are globally threatened because they're endemic to the region. So it's just to emphasize that we do have an obligation, if you like, and there is an imperative, urgent imperative to conserve these species before any more become threatened. And that's all I wanted to say. Thank you very much for listening. And if there's time, I'm happy to take questions now or later on. Thank you. Wojtek, we can't hear you.
Now you can. Thanks again. I just wanted to say that somebody in Europe finally said directly what group of species of priority species should we focus on talking about CWR. So it was very brave that somebody finally said this list. Thanks a lot for this, Sheila. And now I would like to ask Joanna again to check what's up there in our Q&A box.
Yes. Thank you, Sheila. So we have another question from Julian Hosking. He asks, what is the definition of crop that is applied to crop wild relatives? And how do taxa become crops and also become no longer crops?
Thanks for that, Julian. Okay, so a definition of a crop, well, I mean, I guess that's a slightly tricky one. But if you're talking about a broad definition of a crop, you could say that it's anything that's cultivated, anything that's actually in cultivation, which is no longer in the wild. How do taxa become crops and also become no longer crops? Do you mean, okay, so obviously crops come from the wild. So around about 10,000 years ago, humankind started to cultivate species rather than just collecting them from the wild. And this process of domestication is ongoing. There are still a number of wild species which are being brought into cultivation. In my view, as soon as something is taken from the wild and is planted, then it becomes a crop. It's no longer a wild species. Also become no longer crops. Well, I guess once those, if seed escapes from those crops, as they often do, then they occur as wild species again. Then those are wild populations. They may be escapes from crop populations, but they then become wild relatives of those crops. Does that answer the question?
Yeah, well, I think so. It's a difficult question. Then we have another question by Alvina Avagian. I'm sorry, I'm misreading your names, with difficult names. So she asks, this is about crop prioritization criteria. So why criterion on threat status is not the first criterion? And then all crops or taxa have a value. Some crops are of great value nowadays or are used more today. The others have a potential value. They will be used widely as biotechnology techniques are developing. So if we lose any taxon because it's not of high socioeconomic value today, it will be lost forever. So,
okay, thank you. Hi, Alvina. Thank you for your question. Yeah, it's a useful question, and the reason that relative threat status is not the first criterion is that if we apply that as a criterion, we can reduce the number of taxa to a very small number, and then miss taxa which are of utilization potential. So it's important to do this in a logical step-by-step process as proposed in the 2017 paper in Crop Science. And as I said, it is the unique, what distinguishes crop wild relatives from other wild species is their relationship to crops and the fact that they are potential gene donors to crops. So we should look, first of all, at which are the priority crops that are most socioeconomically important, and then conserve the wild relatives of those crops. And if you look at the methodology that I just outlined, you won't miss any threatened species. You will include all of the species which are most closely related and or have potential known use because they've already been characterized. And then in addition, you will include any threatened species which are more distantly related, for which we don't yet have characterization data. Does that answer the question? Maybe there was, oh, the question's disappeared from, oh, there it is. Sorry.
yeah.
Okay, so I think there was another part to the question.
Exactly.
I'm not quite sure if I understood it, but we should also bear in mind, yes, there are many, many crops that we could say are a priority, but the point is that it's unlikely, certainly at this stage, that crop wild relatives would be used in crop improvement programs. Crop wild relatives are used more extensively for, as Nigel mentioned, 15 major food crops, simply because those are the most economically important crops. And to put a lot of resources into breeding using crop wild relatives, that it is not easy to use crop wild relatives in crop improvement programs because of the issue of linkage drag, the introgression of unwanted traits. And so by default, they will be used more for the most economically important crops.
Yeah, thank you, Sheila. I think what Alvina was also saying is that, and I think you have answered, is that sometimes we have species, wild relatives of crops that are very threatened, although they have not a very high socioeconomic value today. And so whether we should also focus on those ones.
Yeah, so that's exactly what I presented with the third criterion. If we apply the third criterion, we make sure that we don't miss any of those species.
Yes. Okay, we have another question by Denise Fudostatni from Poland. So she asks about, what about ornamental plants? We've just finished the crop-wide relative Polish national list, and we included ornamental plants. Can we consider some species as a priority species if they are threatened by extinction, but they are not directly connected with food plants? So this.
Hi, Denise. Thank you for your question. Yeah, this again is a very pertinent question. I want to emphasize again that the prioritization that I've just presented is at regional level. When we prioritize at national level, we may, countries often apply different prioritization criteria. So for a particular country, they may decide also to include ornamental crops, which are, of course, they're not important, of course, for food security, but they are economically important. And so generally, if we take this two-pronged approach that I outlined, we ensure that at regional level, we are conserving the most important crops for food and nutrition security, but those that are related to other economically important crops, such as ornamentals and medicinal plants, would be conserved at national level.
Thanks a lot, Sheila and Joanna, for your help in this short Q&A session. Too short for sure, but remember that you still can write down your questions to Sheila, and also you can ask them later in the general session if you will have some free time. And also, if there are any unanswered questions, we will try to send you a Q&A report after our meeting. And I would like to introduce now our last speaker, Jose Iriondo. He's a professor at the Biodiversity and Conservation Unit of King Juan Carlos University. So he's speaking directly from Madrid in Spain. He has published over 100 research articles, more than 100 publications, book chapters, and more than 80 funded national and international research projects. So great numbers, and also a great project partner. Jose, are you there?
Thank you, Wojtek.
Okay.
Hello, everyone. Can you see my screen?
No, we cannot see. So please try again.
Okay, let me see.
Just a while.
There must be some problem with the PowerPoint presentation. Let me see.
Oh, now it's almost there. Yes, we got it, Jose. And you just need to open presentation. Now it's great. Thanks a lot.
Okay, thank you. So I'm going to talk about the conservation of crop wild relatives in the Natura 2000 network. And as you all know, the Natura 2000 network is the largest network of protected areas in the world. It is the result of the coordination effort among European Union members, having put a strong economic investment to generate it. And in this context, in which we had a great need for the in situ conservation of crop wild relatives, it is logical that we consider the use of conservation infrastructures that are already available, such the case of Natura 2000 network. So the purpose of my presentation is to make an assessment on whether the Natura 2000 network is useful for the in situ conservation of crop wild relatives. And I have structured my presentation in three parts. In the first one, I will assess whether there are European priority crop wild relatives efficiently protected from human disturbance by Natura 2000 network. Secondly, whether some of these crop wild relatives are actively conserved by management actions associated to Natura 2000 network. And finally, to wrap up, just a small discussion on what potential does the Natura 2000 network hold for crop wild relative in situ conservation. So the target crop wild relatives we are going to focus on in this case is the inventory of European priority crop wild relatives that Sheila just presented, composed of 863 taxa related to human and animal food crops. And to begin with the first question on whether there are crop wild relatives efficiently protected from human disturbance by the Natura 2000 network, what we actually need to assess is the coverage of the Natura 2000 network concerning crop wild relative taxon richness and genetic diversity. And to do that, we basically have two approaches. One of them would be to collect the floristic inventories of the different Natura 2000 sites. And the other is to make queries at global biodiversity databases. So concerning the first approach, indeed, many Natura 2000 sites have conducted thorough floristic inventories as part of their management plans. But compiling this information would be an enormous task, considering that there are over 27,000 sites. And it is true that at the webpage of the Natura 2000 webpage held at the European Environmental Agency, there is a file named Other Species, where the Natura 2000 sites provide information on other species other than those contemplated in the Habitats Directive. But these species are species that have been outlined for some reason, mostly because they might be threatened, endemic, or rare. But these files do not contain a thorough catalog of the floristic taxa that are available in these sites. So for practical purposes, in the context of the Farmers' Pride project, we conducted queries in two large global databases: the GBIF, the Global Biodiversity Information Facility, which is a repository of data occurrences that are on wildlife, on wild species, with many data providers worldwide, and the Genesis database, which is also a global database that collects information on seed accessions in gene banks all over the world. So the process we did to collect this data, once we had focused on our priority crop wild relative taxa, was to prepare an R script-based process based on many existing packages. And then with them, we conducted a query on these two large databases. We obtained over 16 million records concerning these species, worldwide occurrences. And then we conducted a series of filtering processes in order to eliminate all these records without coordinates or with low quality geographical coordinates, as well as other aspects, in order to produce a high quality georeferenced database. At one point, we had to join the contents of coming from both large databases, put them in a common template. And finally, in order to focus in the assessment of the Natura 2000, we just filtered the records occurring in the Natura 2000 territory, which is EU 27 plus the UK. And we selected the records dated 1950 and after to increase the probabilities that these records correspond with existing natural populations. So as a result of this, we ended up with a database with over 2.8 million data records for 568 crop-wide relative taxa. That is 2/3 of the taxa included in the inventory of European crop-wide relatives. And they make the largest existing occurrence dataset of European crop-wide relatives. So with this data, we transform it into a shapefile in order to be able to work on a GIS environment. We also obtained a shapefile from the Natura 2000 protected areas containing the polygons of these areas. We joined the two shapefiles in order to obtain the populations of the priority crop-wide relatives that fall within the Natura 2000 sites. The result of this is what you see in this map. We found out that over 400,000 populations of 519 European priority crop-wide relatives are found within the Natura 2000 network. These are the green dots you can see in this map. And they would correspond with the number of priority crop-wide relative populations that are currently passively protected by the Natura 2000 network. This means these amount to circa 14% of the populations recorded in the database, and 91% of the 568 taxa with high quality distribution data in our database. So this is an impressive coverage of the Natura 2000 network. And these populations are found in Natura 2000 sites distributed along all EU 27 plus UK countries. Obviously, not every Natura 2000 site has a crop-wide relative priority taxon. According to our database, 1/3 of Natura 2000 sites hold at least one crop-wide relative priority taxon. And for those sites that contain at least one taxon, the mean number of taxa per site is about 15. We also focused on those crop-wide relative species from the database that had 20 or less populations recorded in the database. And so they might correspond to species with a small distribution areas. And we found out that 72% of taxa in this situation have at least one population in the Natura 2000 sites. So we have recorded these sites that hold populations of crop-wide relatives that are particularly rare and of interest. So to sum up, concerning this first part of the talk, concerning the taxon coverage of European priority crop-wide relative, we can say that there is a very good coverage of priority crop-wide relatives. We should note that this is very conservative data because obviously not all crop-wide relative populations have been recorded by botanical records, and not all existing botanical records have been uploaded into the GBIF and Genesis databases. So there are probably many more populations and many more taxa contained in the Natura 2000 network. But at least we can say for certain that the Natura 2000 network holds more than 590 priority European crop-wide relative taxa and more than 400,000 crop-wide relative populations of interest. Now, we should bear in mind that the focus of crop-wide relative conservation is on preserving genetic diversity. So it is also very important to assess whether the Natura 2000 network covers adequately the infraspecific genetic diversity contained in each target crop-wide relative. If we had unlimited resources, we could just follow a molecular biology approach and conduct genetic analysis on all the populations concerned in order to answer this question. But this is totally unattainable nowadays. So the only resting approach is to follow an ecogeographic approach, which is based on the basis that under particular environmental conditions, some natural selective pressures are exerted on existing additive genetic variation on the populations that prompts adaptive evolution that leads to local adaptation. So there is a link with particular environmental conditions and particular genetic variation of adaptive value. So with these premises, we can combine climatic, edaphic, and geographic data to generate what we call ecogeographical land characterization maps, which consist of ecogeographical units that correspond to particular combinations of climate, soil, and geographic data. And they constitute a proxy for genetic diversity patterns. So we can infer the coverage of genetic diversity through the ecogeographical categories that are represented in the populations occurring in Natura 2000 sites. This is still work we have in progress concerning this Natura 2000 network assessment, but we can already advance that a great part of the genetic diversity assessed through this approach is contained in the Natura 2000 network populations of crop wild relatives that we have identified. So once we are able to conduct this assessment, we will be able to carry out diversity and complementarity analysis based on taxon richness and ecogeographical diversity that will help us identify priority localities for crop wild relative conservation action. The second question I wanted to address is whether there are European priority crop wild relatives actively conserved by management actions associated to Natura 2000 network. And the way we approached to this question was by looking on whether some of these crop wild relatives are species protected by the Habitats Directive Annexes 2, 4, and 5, or by whether some of these crop wild relatives are characteristic plants of Annex 1 habitats protected by the Habitats Directive. So in the first case, we identified 17 crop wild relative priority taxa protected by Annexes 2 and 4 of the Habitats Directive. And in the second case, we searched in the interpretation manual of European Union habitats whether, defining each particular habitat, there are these characteristic species, plants that are the structural species that conform these habitats. So we can assume that the conservation actions that are being taken place in the Natura 2000 sites to conserve these habitats are actually always in favor of maintaining and conserving these structural plants, these characteristic plants. And so we found, when we cross-checked these characteristic species, that 83 of the 233 protected habitat types contained at least one European priority crop wild relative. And in total, a total of 84 priority crop wild relative taxa are characteristic plants of these habitats. So we have a complete list of them, and we know for which habitats they are characteristic species. So as a whole, we have identified 17 crop wild relatives in Annexes 2 and 4, 84 as characteristic species of Annex 1 habitats. 3 of them are found both in A, B items. So in total, there are 98 crop wild relatives for which we can say that are nowadays actively conserved by management actions associated to Natura 2000 network. So to wrap up, what potential does Natura 2000 network hold for crop wildlife in situ conservation? First, we have seen that Natura 2000 holds great taxonomic and possibly genetic diversity coverage of European priority crop relatives in terms of passive conservation. Moreover, a relevant number of European priority crop relatives, 98, are actively in situ conserved by standard Natura 2000 network management practices. So we can conclude that Natura 2000 network is an essential foundation for in situ crop relative conservation in Europe. Should be noted that now substantial further investment is needed to apply to implement this conservation. Just recognition of these valuable plant genetic resources in each Natura 2000 site, identifying the crop relatives that occur in each site, and taking occurring crop relative populations into consideration when generating or updating existing protected areas management plans. So thank you very much. This is all. And if you are interested in getting additional information, I'll leave you these links where you can obtain more information on the project, on the network initiative, where there is a survey you can take pressing this button. And you can also access a briefing document on the presentation I just gave, which is available either through this link or in this place in the web page. Thank you very much.
Thanks a lot, Jose. It's really very important to relate CWR conservation with existing networks, big networks such as Natura 2000. And now we've got just 5 minutes for a kind of final questions. And just remember that still you can write down this question in Q&A. Probably quite a lot of them will be unanswered, but we'll just send them to panelists, asking them to help us with the report from the session. But still we can ask something like 1 or 2 questions, and later we'll have a kind of very short summary of our meeting. So, Joanna, what about Q&A box?
Yes, so we have a question from Luigi Guarino. So he asks whether there are at least some populations of some species in all European countries.
Yeah, as you were able to see in the map with the green dots, we were able to see that there are European crop relative populations found in protected areas of the Natura 2000 network distributed along all EU 27 plus the UK countries. So they are found in all countries.
He also asks whether you could remind him, because he missed it, how many of the priority crop wild relatives percentage are not found in Natura 2000 sites?
Well, see, we can only say that for the 500 and some priority crop wild relatives for which we obtain high quality distribution data in our database. So as I said previously, 91% of these taxa are contained in the Natura 2000 network. So that just leaves 9% of the taxa of our database that are not found in the Natura 2000. But that's, I should say that this according to our records. As I said, the results I am showing are very conservative data. So even for this 9%, we could do further research, and I wouldn't be surprised that we ended up finding at least some of them in the Natura 2000 sites.
Thank you, Jose. I think
Sheila is raising her hand.
Yeah, exactly.
Yes, thank you. No, I just wanted to also say that in response to Luigi's question, that we know from EuroPlusMed plant base, which is the basis, the taxonomic standard for our inventory of priority crop wild relatives, we know that the priority taxa occur in 44 countries. And also just to say that this work that Jose has presented today is specifically on the occurrence of crop wild relatives in the Natura 2000 network. But that isn't to say that there aren't also crop wild relatives occurring in other types of, in other protected areas outside of Natura 2000. So for example, privately owned protected areas as well are important. But that is an extension of the analysis that we're undertaking, is also to look at other protected area types as well.
Okay, great. And a final question, very final.
Yes, so CEO Van Hinten from the Netherlands, he asks, how does protection in the Natura 2000 network relate to the concept presented by Nigel?
I think he means to the crop relative concept.
Perhaps, Theo, maybe you can elaborate a little bit more.
Maybe I can try answering that question.
Yes.
Or is the question specifically for Nigel?
No, it doesn't refer to any of the presenters, so I think you can answer, yeah.
So, Theo, hi Theo, if you're referring, I'm assuming you're referring to the CPGR concept for in situ conservation of crop wild relatives in Europe. That concept doesn't specifically necessarily mention the Natura 2000 network. I can't remember exactly, possibly it does. It relates in the sense that we're establishing, what we want to establish a network for in situ conservation of crop wild relatives, regardless of where those sites occur. And the Natura 2000 network is already established. All of the EU countries have Natura 2000 sites. And so it's an obvious initial vehicle for taking forward the in situ network for crop wild relative conservation. I don't know if that answers the question, or Nigel, if you have anything to add.
No, I think that when we originally wrote that document, we were thinking that, you know, our thoughts on the subject have changed over time. And initially, I think when we were writing that document, we had no clear idea which network of protected areas we would take forward. But now, having thought about it for several years, it seems to me the only option for taking forward in situ conservation in existing protected areas in Europe is jointly with the Natura 2000 network. There isn't another option, because they are the protected area network that has the matrix of sites across the whole of, well, the EU plus the UK. And perhaps outside of that, we could add other kinds of protected areas in to make a really, truly European network. But the basis of the network, I think, will be the Natura 2000 network. And so that's why I'm so pleased to be able to have this session to talk about it and try and establish a firm link between our interests in agrobiodiversity conservation and the interests of the Eurosite and the Natura 2000 network in terms of conserving agrobiodiversity.
I can see that there's still quite a hot discussion in Q&A box. So I've got a proposal that I will officially close slowly our webinar, but I will leave a room for 5 minutes or so that you can still write your questions and ask something. So just to keep our time, from my side, I would say that thanks a lot to all of you, all our panelists and a great group from Farmers' Pride project. And it's really a great pleasure for Eurosite and our secretariat to cooperate with you, because the aim of our network is to build a bridge between scientists, experts, and site managers. And you are exactly filling this gap between them. So here on your screen, you can still can see some links, and just after our meeting, you will get a report with links and a place that you can download the recording from our webinar, and also within a week time or something like that, we will prepare the detailed Q&A report with all of these questions that were unanswered during the meeting. And that's it from my side. Somebody from Farmers Pride coordinators would like to say something important for all?
Well, I think I just made the point that this has been very useful for us in establishing a direct link with the people who actually manage the protected areas, and to emphasize, if we are going to achieve our goal, our long-term goal of setting up in situ conservation of crop wild relatives and land races across Europe, then the only way to do it is in collaboration with protected area managers. And so we're very open to that kind of collaboration. And as I said earlier, and Jose also stressed, we have this current survey we're taking. So if you're managing a protected area and you have either diversity of crop wild relatives or land races within that area, then we'd like to hear about your site for potentially including it in the network when it is launched next April.
Yeah. Thanks a lot, Nigel. I would like just to add that I've seen on our participants list that during our webinar, there were more than 90 people joining us today. So it is a really great group to get this knowledge from you, and I'm really happy that you've been with us for this quite a long time. So now I'm closing the official recording, but still, if you've got something in mind, just you've got the last chance to write down the question in Q&A. Thanks a lot, Jose. Thanks a lot, Joanna, Sheila, Nigel, and also Jelke from our secretariat was really very helpful in preparation of this webinar. And I cannot see her, but probably you can.
Could I just say thank you very much to Eurosite for hosting this webinar. It's been a great to have you in this project and a great opportunity. So thank you very much to Jelka and Wojciech for the collaboration.
Very welcome, and thank you as well.
Thanks a lot.
And thanks to everyone for attending.
Yeah. So still Q&A box open. Last 5 minutes to write something down. Thanks a lot to all of you. Bye-bye.
Thank you. Bye. Bye.